this tartar, which we in Germany use to remember the structure of carbohydrates. Actually, it's very well known in Germany, so I tried to show you this little song. Okay, so I hope the volume is okay now. Song worked before, after the channel. Do I understand why I'm working here? But they fixed it actually in the meantime. Last time it wasn't my fault, I guess. I hope you remember the Tütetana or something similar. You'll see in a little while when I talk about derivatives of carbohydrates and such that it really makes sense to know the structures of molecules like glucose. So again, if that doesn't work for you here because you don't speak German or whatever, come up with something. So, however, so we talked about the straight chain form of carbohydrates, glucose in particular, and some related molecules. And then I told you, or reminded you again, I should say, that in solution, these molecules, at least if they have, let's say, 5 or more carbons in their structure, have a tendency to cyclize. We talked about this cyclization process. We talked about the process in which the anomers that can be formed in the cyclization can be interconverted into each other in this process called mutarotation. And what I would like to discuss briefly now is how we represent the cyclic structure of carbohydrates and how we can relate this cyclic structure to the open chain structure, the Fischer projection. So obviously here we have the cyclic structures, and depending on whether we have a 6-membered ring which comprises the oxygen which attacked the anomeric carbon in the cyclization process, and 1, 2, 3, 4, 5 other carbons to make a 6-membered ring, or whether we have a 5-membered ring that comprises 1 oxygen that participated in ring closure and 1, 2, 3, 4 carbons. We, based on the similarity to these 2 organic chemicals, which are pyrin and furan, call these cyclic structures pyranoses or furanoses. Okay, so if we include the ending pyranose in a name like glucopyranose, we know it's a cyclic structure, 6-membered ring of glucose. Similarly, if we have furanose in such a name like here, fructofuranose, we're dealing with a 5-membered ring. Okay, so all this should be familiar to you. When we have these projections, all those projections, it's somehow implied that we are dealing with planar ring systems, which, as you should know, is actually not the case. However, if we focus here on pyranoses, the oxygen and the carbons 2, 3, and 5 actually typically lie in one plane, and carbons 1 and 4 are out of the plane, and how they are out of the plane I'll show you in a moment. So remember that the positioning of the hydroxyl group at the anomeric carbon depends on how the ring closure occurred, meaning whether the attack of the oxygen came from above or below with regard to the planar structure of the aldehyde functionality. Okay. So how do we relate the Fischer and the Horvath projection? See here that everything, all the hydroxyl groups, I should say, that are linked to chiral centers in a carbohydrate and point to the right in the Fischer projection actually point downwards in the Horvath projection. Okay, and everything that points to the left here goes up in the Horvath projection. Now this is not true for obvious reasons if you think about it for the anomeric carbon, where again the position of the hydroxyl group and the hydrogen for that matter depends on whether the attack came from above or below the plate. Okay. But all the functional groups besides the ones at the anomeric carbon follow this rule here. So if we say here, oops, tar, and we look at the positioning of the hydroxyl group in carbon 2, 3, and 4, this one will actually, this oxygen will participate in the ring closure. We see, okay, tar, okay. Now, occasionally I get the question why the hydroxymethyl group in these sugars, actually these pyranoses actually points upwards. Now if you build a model, you actually see that it points upwards. You can also deduce this from a two-dimensional representation, which I'm not going to do here. But this is actually, or this actually applies to D sugars. So in D sugar, and by convention we would represent the D sugars in such a way, or any sugar actually, that the oxygen that participates here in the formation of the ring is in the upper right corner, and if we're dealing with D sugar, the hydroxymethyl group points actually upwards in this ring structure. So remember that. We'll see this again in a moment. Okay. So as I said, while the Horvath projection implies that we're dealing with planar ring systems that would involve all 6 atoms in a pyranose, this is not the case in reality for steric reasons and energetic reasons. So actually, a couple of conformations can be assumed by these ring systems, shown here using cyclohexane as the classical model system to show such stereochemistry in ring systems. But we'll see a representation of a pyranose in a moment where the oxygen would actually be placed here or here. Now these are, let's say, the more prominent conformations that could be assumed. Here, the boat conformation, and we see that if we have substituents, especially bulky substituents in these positions, that we have some steric clashes, and for that reason, this is not thermodynamically favorable. More favorable is such a chair conformation, where our substituents at the ring carbons are actually placed either in equatorial or axial position. As you may remember from organic chemistry, if we have bulkier substituents, it's sterically, thermodynamically more favorable to place these substituents actually in equatorial position. Okay, so we minimize steric interference with other substituents. So let's take a look how this would actually look like for a pyranose. So here we are dealing with glucopyranose, and we see that we can actually form 2 different types of chair conformations. Again, the oxygen and these 3 carbons actually lie in one plane. The carbon 1 and 4 actually are out of the plane, and we can have 2 different conformations that can be interconverted into each other, which we can refer to as 4C1 or 1C4. So the superscript number here implies that this carbon actually is above the plane, and the subscript implies that this carbon would be below the plane. Okay. Now if we look at this molecule here, glucopyranose, we are actually dealing with what anomer here? Which pyranose? Glucopyranose anomer. What possibilities do we have?
We just be alpha and beta.
Okay, so it's either alpha or beta. What is it? look at the hydroxymethyl group, carbon number 6, and the hydroxyl group, they are both up, right? And certainly not down, so they are on the same side. So what are we dealing with then? Beta. Okay. So I told you that if we place our more bulkier substituents in equatorial position, that we minimize steric interference. This has happened here in the beta anomer, right? So what would the alpha anomer look like? Excuse me. Yeah, so we would switch these two. So this hydroxyl group would actually be placed here, down here, would be in axial position, and the hydrogen would be here in equatorial position. So then these two hydroxyl groups here, if we had one here, the alpha anomer, would be relatively close, and for that reason, the beta, both these hydroxyl groups, the one at the anomeric carbon number 1 and the one carbon number 2 are in equatorial position, is thermodynamically more stable. Okay, and we see this in aqueous solution because about 2/3 of the molecules are in beta anomeric conformation. Okay. So obviously also the 4C1 is somewhat more stable than the 1C4 conformation. So far we've only talked about underrivetized monosaccharides. There are plenty of different derivatives of carbohydrates, of monosaccharides, and so on. A few are shown here. So here obviously we have an underrivetized glucose. Here we have replaced the hydroxyl group at carbon number 2, excuse me, with an amino group, and we actually produced an amino sugar. As the name implies, we just add amine to the stem of the word here and call it glucosamine. Now similarly, if we added amine at carbon number 2 in galactose, we call it galactosamine, and mannose, we call it mannosamine, and so on. Now the amine often enough is acetylated, so we would call this N-acetyl, N to show that the acetyl group is actually linked to nitrogen, and acetyl glucosamine. We've already seen examples of phosphorylated sugars. We'll see plenty of phosphorylated sugars in this class. As we continue our discussion of metabolism, here's glucose 6-phosphate. We talked about this molecule already. Now there are a bunch of carbohydrates that have carboxylic acid functionality, so they are acidic. Here are some shown that we would find easily in bacterial cell walls, for example. Here are some glucuronate and gluconate, which we're going to discuss in more detail in a moment. So this is an intramolecular ester, a lactone that can be formed here from such uronic acid. Here is N-acetylneurominic acid, which also has carboxylic acid functionality. This is a participant of oligosaccharide that we find on the surface of cells, where it also contributes negative charges that overall make the cell surface actually negatively charged. What I would like to spend a little more time on, for the time being actually, is these two sugars, not because they are particularly important, but we see the difference between D and L sugars. So as I implied previously, D sugars are more prevalent in nature, does not mean that there aren't L sugars also found in nature. Here are two that are found in nature, L-fucose and L-rhamnose. Now if you look at these structures here, these Horvath projections are written the same way, following this convention that we've seen previously for glucose, for example, where the ring oxygen is found in the upper right corner. However, the functional of the let's say, more prominent substituents at carbon number 5, in this case, is not a hydroxymethyl group, but just a methyl group, points downwards, which shows us that this is an L sugar, okay? And I have added, actually, some other names for these that you could give these molecules. So L-fucose is actually nothing else but 6-deoxy-L-galactose. 6-deoxy shows us that there is no oxygen at carbon number 6 anymore. That's why we don't have a hydroxy methyl group anymore, but just a methyl group. And then, besides this difference, it's simply L-galactose. Okay, so here we have at least an L-galactosamine, a D-galactosamine, excuse me. And if you look closely enough, both are in the beta conformation, they are the mirror images of each other. Okay, disregard the missing hydroxyl group here, carbon 6 in this fucose, and disregard this amine that we have in the galactosamine here. But otherwise, everything that was pointing downwards in this D sugar is now pointing upwards in the L sugar, because oxygen and the 5 carbons serves as a mirror plane. Okay, so we project everything pretty much through the corresponding point in the ring system, and so if we form the mirror image of this molecule, well, let's go to this one, which is the L, and we project this hydroxyl group through this point, it has to point upwards in the mirror image in enantiomer, which would be the D sugar. Okay. Similarly, if you look at the structure here of alpha L-rhamnose, it's the same as 6-deoxy-L-mannose. If you compare this with this structure, you'll see the same thing happen. Okay, so that's just to give you an idea about how we can relate these structures to each other. It's not black magic. Okay, it's actually quite simple once you know how to do it. Okay, let's take a look at some important acidic sugars, sugar acids. So aldonic acids actually are carbohydrates where the aldehyde functionality at carbon number 1 has been oxidized to a carboxylic acid. Now this can be done in the lab quite easily with relatively mild oxidizing reagents like copper 2+ solutions. So we always have coupled redox reactions. Remember that if the aldehyde here is oxidized to the acid, we have a reduction of the copper 2+ to copper 1+, okay? And we have converted glucose, the aldose, to a so-called aldonic acid. And we imply this by calling glucose then gluconic acid, or to indicate that we are dealing with the ionized form, gluconate. This redox reaction can also be utilized in tests like Feeling's test or Tollens reagent to actually test for the presence of aldoses. Often enough, especially biology students tell me that tests like Feeling will also be positive with ketoses. However, what you should be aware of is that this is an experimental artifact, which is based on the fact that you have base present in Feeling's reagent, which converts the ketose, fructose, to aldose, okay, and then your aldehyde functionality is actually oxidized to the acid, and your copper present in copper sulfate is actually reduced to copper 1+, and you have a change in color, okay, maybe even precipitation. So this tests for aldoses. Okay, one term that you should be aware of and should remember, I hope you do, is the reducing power of sugars. So what this means is that we are dealing with carbohydrates that can still be oxidized and participate in redox reactions, so they can reduce other molecules, just like our aldehyde here, as it was oxidized, reduce the copper 2+ to copper 1+, okay? And this nomenclature of reducing power or not having a reducing power is important, as it also allows us to distinguish the ends of polymers of carbohydrates and distinguish between a reducing end and a non-reducing end. Okay, we'll come back to that in a little while. Another important product of the oxidation of acids are uronic acids, where there are others also. Bones are oxidized, for example. In uronic acid, carbon number 6 has been oxidized, and we form molecules like glucuronic acid, galacturonic acid. Now, glucuronic acid we'll see again when we talk about the degradation of heme, because glucuronic acid is utilized there and also in other reactions to make a very hydrophobic intermediate in the breakdown of something. Here we're talking about heme, more hydrophilic and water soluble to facilitate its secretion from our body. Okay, and so glucuronidation reactions are important, or are the quintessential, really, or not quintessential, but very important so-called phase 2 reactions in detoxification processes. So if you take up also some hydrophobic compounds like herbicides or something like this, your body actually will deposit them, since they are hydrophobic, to a large extent in your fat tissue, because it's very hard to secrete them. Anything that we want to secrete, we really have to make sufficiently water soluble. We are actually 60 to 70% or more water. So in order to get rid of stuff, we have to have the molecules dissolved in some aqueous environment. And in order to make hydrophobic molecules more water soluble in detoxification reactions, we can first introduce more hydrophilic groups, or groups that are amenable to further derivatization, for example, to attach glucuronic acid. And then if you have a sugar which has lots of hydroxyl groups, we actually increase the hydrophilicity of these molecules and make it easier for these molecules to be secreted. We'll see an example for that when we talk about heat degradation. Okay, now let's talk about oligomers and polymers of saccharides. Disaccharides, there are only a couple of disaccharides that are relevant, really. They are shown actually on this slide here. These can all be called glycosides, and a glycoside, let me jump back to this slide here, is simply a derivative of the hemiacetal or hemiketal that is formed in the cyclization process, the cyclization of a straight chain sugar. Okay, so remember, if we simply cyclize glucose, for example, we would have a hydroxyl group here, but I indicated last time that this anomeric carbon is quite reactive and can easily be attacked by other nucleophiles and can be derivatized. And the derivative we can refer to as a glycoside. And the bond between the sugar and this molecule that participated in this derivatization is called a glycosidic bond. Often enough, it's via an oxygen, then we could refer to the bond as an O-glycosidic bond, but we could also have a nitrogen attack here, and then we would refer to this as an N-glycosidic bond. If you have, for example, a ribose attacked by nitrogen number 1 in pyrimidine base, a nitrogenous base, for example, you would have an N-glycosidic bond, or nitrogen 9 in a purine base, you would have an N-glycosidic bond. Here, we have an O-glycosidic bond simply to a methanol. Never mind, but this is still a glycoside. Now, this is not particularly important for physiological systems. What is more important actually in physiological systems are other glycosides. I mentioned bonds to nitrogenous bases already, but what we're looking at here, and we're still talking about saccharides, polygon and polysaccharides, is the nucleophilic attack that actually occurs from the hydroxyl oxygen of another sugar to this anomeric carbon of the first sugar. So if we have a glucose here and a glucose there, we have the anomeric carbon and the hemiacetal here in this first sugar. If we have the hydroxyl oxygen of carbon number 4 in this example attack this anomeric carbon, we have a condensation reaction. Water is released here, and we form a covalent bond that looks kind of like an ether bond, but it's not an ether bond. It's an acetal that is formed here. Okay, so if we derivatize this hemiacetal in such a nucleophilic attack, we form an acetal. So here we have linked covalently two sugars, two glucose molecules, via a so-called alpha 1 to 4 bond, and we formed the disaccharide maltose, which isn't really formed in this way in nature. It's a degradation product of starch. Okay, but it could form this in lab if you want. Now let's talk a little bit about nomenclature here with regard to the ends. So in all polymers that we find in nature, we have directionality, and we can distinguish the two ends, also in carbohydrates. If you look at these structures, we still have a hemiacetal here at this end, because the anomeric carbon or the hydroxyl oxygen here at the anomeric carbon did not participate in any further derivatization. So this hemiacetal can still be quite easily opened up to regenerate the aldehyde functionality, and then the aldehyde could participate in a redox reaction, could be oxidized to carboxylic acid, reduce something else. So this molecule, as shown here, while this hemiacetal still possesses on this side reducing power. Therefore, we can call this end the reducing end. Here on this side, we do not have a hemiacetal or anything like that anymore. So we do not have reducing power. So such polymers or sugars have two ends. Okay, never mind. So what's the convention? How to describe these molecules? We write the compound with its non-reducing end to the left, so we know what the non-reducing end is. Then we show the configuration at the anomeric carbon. So this means, is our bond going upwards with respect to this hydroxymethyl group here, or is it going downwards? So it's going downwards. So the bond here and the reference point here, opposite sides of the plane. So therefore we have the alpha constellation, alpha configuration. If it would go upwards, they would be on the same side, and we could call it beta. Then we actually can include furanosyl or pyranosyl to show if we have 5-membered or 6-membered rings. And then we also show where the bond occurs by giving the number of the carbon. Let's go back here, carbon number 1, and connect it with an arrow to show what the other participating carbon participating in this bond is. So this is an alpha 1 to 4 bond. Okay. So here are just the important disaccharides that we find in nature. Hope you're familiar with these structures. So maltose, I mentioned already, the degradation product of starch. There is a shorthand notation which utilizes the 3 letters abbreviations or synonyms or names of the carbohydrates. So GLC stands for glucose, obviously. So if we have glucose linked to another glucose via an alpha 1 to 4 glycosidic bond, we're dealing with maltose. If we have galactose linked via beta 1 to 4 bond to glucose, we're dealing with lactose. If we have a glucose linked to a fructose, okay, we're actually dealing with sucrose, table sugar. However, what you have to notice here is that both anomeric carbons in the 2 sugars have been involved in the formation of this bond. So you can either write alpha 1, 2, and that should be actually a continuous line here, to 2 beta or fructose, or you could reverse this because it's the same, okay, because we do not have a non-reducing end that we could write on the left. And similarly, in trehalose, actually we have both of the anomeric carbons involved in the formation of the glycosidic bond, meaning we can indicate this with such a double arrow. The bond is alpha 1 to alpha 1, regardless of how you write it, it's the same thing. Now let's talk about higher polymers of polysaccharides. There aren't any underivatized 3 oligosaccharides that occur in animals. There are some in plants. I'm not talking about plants here. So there are oligosaccharides, as you know, as parts of conjugates with other biomolecules, be it lipids or proteins, and we'll talk about those also in this class. So if we just talk about polymers of sugars, we can move on to polysaccharides, which we can also call glycans, and all I will talk about here in this class, in this review, are just so-called homopolysaccharides, which means that we use the same building block over and over to form a chain or branch chain of this molecule. You may remember from 472, I hope, that there are also heteropolysaccharides, where disaccharide units are actually used in the polymerization process. But here we're just talking briefly about polysaccharide. So when we talk about homopolysaccharides, any polysaccharide, what do we have to take into account? First of all, what's the nature of the building block? So which molecule has been polymerized over and over? In the case of heteropolysaccharides, these disaccharide units. Then if we compare different polysaccharides, they can differ with regard to the overall lengths of these chains. What is of great importance actually for the functions of these and the properties, structural properties of these molecules is what type of bond we have. And what I mean here is the orientation of the bond, whether it's an alpha glycosidic or beta glycosidic bond. And then also whether or not we have branching points. Okay, so simple one is amylose, where we just continue pretty much the polymerization that we saw already occur for maltose, bringing in more glucose building blocks and link these building blocks via alpha-1 to 4 glycosidic bonds to the growing chain. And then we form a molecule that again has a non-reducing end and a reducing end, which forms such a left-handed spiral and can have up to 5,000 residues. So this is amylose. Again, it's a relatively simple structure. It's one of the two components that we find in starch. Okay, so this is produced in plants. We can take it up with our diet and break it down. Okay. The other component of starch is amylopectin, where we also have these alpha-1 to 4 glycosidic bonds. But then we have every 24 to 30 residues in these alpha-1 to 4 glycosidic chains with branching points, where the anomeric carbon, so the formerly reducing end of one alpha-1 to 4 chain, has been attacked by the hydroxyl group, hydroxyl oxygen, linked to carbon number 6 in another chain. And we introduce these alpha-1 to 6 branching points, and this happens every 24 to approximately 30 residues in amylopectin, again in starch. And then we can form structures that are actually quite huge and can comprise up to 1 million residues, so big molecules. Okay, both amylopectin and amylose have some water solubility. Okay, water solubility isn't the greatest, but since, as you may remember from other classes, these hydroxyl groups here are still approachable by water molecules, we can hydrate these, and this actually confers some water solubility to these molecules. However, they're actually stored as granules in plants, in chloroplasts and myeloplasts. Chloroplasts is, as you know, through photosynthesis, the location where sugar is being produced. And we also, animals can also store starch, well, no, not starch, we can actually store, we don't store starch, we store the corresponding molecule, corresponding or similar to amylopectin, which we call glycogen, and has a few differences comparing to amylopectin, but we store this reservoir form or the storage form of carbohydrates also in the form of granules, and we store these granules in particular in the liver, but also in skeletal muscle, actually more in skeletal muscle, but the glycogen, or the sugar that is stored there in skeletal muscle, actually serves as an energy reservoir for skeletal muscle, while the sugar stored in the form of glycogen in the liver actually is important for the sugar homeostasis in our body, because the liver actually can break down this glycogen, release it to the bloodstream, and then provide glucose to other tissues that need the glucose as an energy source. So what's the difference of glycogen compared to amylopectin here? We saw that amylopectin can have up to 1 million building blocks, and the glycogen is much smaller, up to about 50,000 residues. And then also it's more highly branched, while in amylopectin we had branching points every about 24 to 30 or 32 residues. We actually have branching points in glycogen every 8 to 12 residues. Excuse me. Okay, I said before that the orientation of the glycosidic bond is very important because it has a dramatic effect on the structure of these polymers. And we see this if we look at cellulose. It's also made of glucose, as we may remember. And the only real difference, if we disregard potential differences in the chain length, is the orientation of the glycosidic bond. While in amylopectin, okay, which formed these spirals, we have alpha 1 to 4 glycosidic bonds. In cellulose, we have beta 1 to 4 glycosidic bonds, and therefore we form individual strands that are relatively straight and can interact with other such strands in 2 dimensions through hydrogen bonds and 3 dimensions also through hydrogen bonds. So the hydroxyl groups participate in hydrogen bonding, which greatly stabilizes these structures that are formed. As you know, cellulose is found in plant cell walls and is very important for the stability of these cell walls and then also wood. Okay, so just the orientation, simple thing to do, right? If you had to build molecules and would want to change properties of molecules dramatically, would you ever think of something like this? Not before trying, probably, okay? So another very stable molecule that is also based on such beta 1 to 4 glycosidic bonds is chitin. Okay, chitin, as you may know, utilizes N-acetylated glucose, so we are dealing with an N-acetylglucosamine as a building block, so there's a difference to cellulose. But we also utilize a beta 1 to 4 glycosidic bond formed chains that are very large. I haven't found any numbers here, to make a structure that we can use to produce exoskeleton of crustaceans and insects, for example, where we find chitin also in the cell walls of most fungi and algae. Okay, so chitin, just think of the exoskeleton of a bug, for example. Quite stable. Okay, so this table just summarizes the major properties of these polysaccharides. Again, you should be somewhat familiar with this stuff. We will talk in particular about glycogen and then to a lesser extent also starch. We're not going to see cellulose and chitin again in this class. So what we're going to do now is start to talk about how carbohydrates can be broken down for energy production. So we'll talk about glycolysis. And why do we start with glycolysis? I've indicated this before because glucose is actually the major energy source of most organisms. It's relatively rich in energy. I gave you this number before. It's something that you may keep in mind somehow. Not the exact number, but to get an idea, minus 2,840 kilojoules per mole under standard conditions if we completely oxidize glucose. So this is quite a bit of energy. We'll see that we don't utilize all this energy when we break it down, and then it also depends on whether we break it down under anaerobic conditions or aerobic conditions. So we'll see that there's a huge difference with regard to the efficiency. But why do pretty much all textbooks and all biochemistry classes that talk about metabolism start with a discussion of glycolysis? I guess almost all at least. Because glycolysis was also the first metabolic pathway that was elucidated, which happened in the 1930s and 1940s. And many of the experimental techniques that were actually used and developed to study glycolysis are actually still in use and provided the basis for, let's say, more advanced techniques. Okay, so glycolysis is of extreme importance, has been of extreme importance for biochemistry as a field also. So, as I said before, glucose holds a central position in metabolism, not only to provide energy when it's broken down to pyruvate here in this process called glycolysis. So we'll talk about this process now, but glucose, as we saw previously, can also utilize as a building block for the formation of storage molecules like glycogen, starch, disaccharides also. can also participate in the formation of extracellular matrix and also cell wall polysaccharides. And then, and that's something that we shouldn't forget, glucose is also pretty much the parent compound of all other carbohydrates, including also pentoses. And glucose is, this hexose is actually converted into pentoses in this pathway called pentose phosphate pathway, where we produce ribose 5-phosphate. Now, I probably won't have time to talk about this pathway in this class, but if I don't forget at the relevant point, I will encourage you to read up on this in the textbook. Maybe we'll have a student presentation, but I don't think so. Okay, so we'll focus here on conversion of glucose to pyruvate in glycolysis, and just as a reminder, so that you keep, let's say, stay excited here when we talk about this stuff and don't get despair. These are the things that we should be looking for when we talk about metabolic pathway. We should look for the chemistry behind these steps where these intermediates are interconverted to each other. We should take a look at the mechanisms of the enzymes participating in these conversions. We should try to understand the mechanisms that actually control the flux of metabolites through a particular pathway. So which are the enzymes that are being regulated, how are they being regulated, and we also get an idea about which are the regulatory steps when we look at the energetics of these steps, because if we want to look for regulatory steps, we should look for exergonic reactions, irreversible reactions. Okay, so we can break down 10 steps of glycolysis. By the way, glycolysis occurs in the cytosol of eukaryotic cells, cytoplasm. Can break down these 10 steps into 2 5-step parts. Now this is a man-made definition of distinction, but nevertheless it helps us to approach these 10 steps. And if we look at these first 5 steps, we can summarize them under the name preparatory phase. So obviously we prepare some molecules for something that then follows the so-called payoff phase. Okay. So if we just take a quick glance at this slide here, which summarizes the first 5 steps, and again we'll take a more detailed look at this in a moment, you have to ask yourself why there are 2 steps actually where ATP is consumed if this whole pathway is all about energy production. This is somewhat counterintuitive, but then I would like to point out that we are still in the preparatory phase here, and we prepare in this preparatory phase some molecules, some intermediates, to be easily converted into other intermediates and to produce energy, more energy ultimately than what we invested here in the preparatory phase. Okay, and if we look at just take a quick glance actually at the payoff phase, we see that there are 2 steps again where ATP is produced, and actually we'll see that everything here is multiplied by 2. So as a matter of fact, the preparatory phase for 1 molecule of glucose that is being degraded in glycolysis, we produce 4 molecules of ATP. We invested 2 molecules of ATP in the preparatory phase, so the net result with regard to ATP production would be 2 molecules of ATP. But then we also produce 2 molecules of NADH, and NADH is also an energy carrier, as I pointed out previously, the reduced form of nicotine amide adenine dinucleotide. But this energy can only be utilized under aerobic conditions when we actually perform electron transport and oxidative phosphorylation in the mitochondrial matrix. Okay. Under anaerobic conditions, this energy stored in the form of NADH for the time being is lost, and the net result of the breakdown of one molecule of glucose under anaerobic conditions would simply be 2 molecules of ATP. Now, if you think about it, under standard transformed conditions, what are 2 ATP molecules worth? How much energy? ATP to ADP, provides how much energy? 30.5 kilojoules per mole times 2 is? 30.5 times 2, what is it? 60 more. Under anaerobic conditions, breakdown of 1 molecule of glucose, which is worth how much? How much did I say before? You have to be quicker, you have to think. How much did I say is glucose worth? Just 3 slides ago I showed you that already. 2,840 kilojoules per mole. That's the worth of a molecule of glucose. Or per mole, let's say it's related to mole. Okay, 1 mole of glucose, 2,840 kilojoules per mole. Now we see that if we break down glucose under anaerobic conditions, 2 molecules of pyruvate would produce ATP worth 61 kilojoules per mole. Do you call this efficient? No, efficiency is about 2%. Okay, but everything's relative. It's good enough to keep us alive. Okay, and us and many other organisms, or muscles, other tissues that actually utilize this degradation of glucose under anaerobic conditions. But other organisms and other tissues actually do just that. Okay, and good enough for them. So everything's relative. In nature and biochemistry, everything's relative. But we'll see that under aerobic conditions, the efficiency is actually quite a bit higher, a factor of 15 to 16. Okay, so let's take a closer look at the individual reactions that make this 10-step pathway. For those of you who took my 470 class, we talked about this in 470. I'll take a somewhat closer look at some of these enzymatic mechanisms and so on. Okay. So, glucose is converted to glucose 6-phosphate. We already talked about this reaction when we talked about bioenergetics. It's a simple phosphorylation of our carbon number 6 here to introduce such a phosphoryl group. This would be this group and form phosphate here. Okay. So the source of this phosphoryl group here is ATP. So here is the first step where ATP is actually invested. And we form ADP, and the enzyme that catalyzes this overall hexagonic reaction, which is hexagonic not only under standard transform conditions, but also under physiological conditions, is catalyzed by a class of isozymes that we can call hexokinases. Okay. So isozymes, as you may know, are enzymes that catalyze the same reaction, but they coded in different genes. And there are a couple of hexokinases that pretty much can catalyze the same reaction here, phosphorylation, glucose to glucose 6-phosphate. Now, when I talk about hexokinase, I usually refer to a hexokinase that we would find, for example, in muscle tissue, which would be typically hexokinase 2. We also find hexokinase 1 in muscle, but hexokinase 2 is more prevalent. And when I talk about the other important isozyme of hexokinase, hexokinase 4 in this class, I will refer to it as glucokinase. Okay? So keep this in mind. When I talk about hexokinase, I mean the one that occurs in tissues like muscle. If I want to specify that I'm talking about the isozyme that we find in the liver, I will call it glucokinase. Okay. Now, why do I make this distinction? Because this muscle hexokinase and other hexokinases and this hexokinase 4, so glucokinase, are somewhat different with regard to their specificity for the substrates, also with regard to the regulation, and also with regard to their Km values. And we'll talk about this in a bit more detail when we talk about regulation of glycolysis. Okay. So as this single arrow pointing from left to right implies, and this number here, which is relatively negative, this is an irreversible reaction. Okay, and I already said that hexokinase and glucokinase are regulated. So this is our first regulatory point in glycolysis. Now, often in textbooks you also find that there is some rationale behind making this the first step, because glucose once it's phosphorylated and carries negative charges, can't leave the cell anymore. So once it's taken up by appropriate transporters and is phosphorylated in the cell, it won't leave the cell anymore. Okay. So this is just how the nucleophilic attack here occurs of the hydroxyl oxygen with carbon number 6 to the alpha phosphorus here, or the gamma phosphorus, gamma phosphorus or gamma phosphate here in ATP. Now ATP, as you may know from 472, is complex with magnesium. This helps here to shield the negative charges and facilitate this nucleophilic attack on this partially positive phosphorus here. We form a bond between this oxygen and this phosphorus and cleave this bond and form ADP and the phosphorylated glucose 6-phosphate. Would you mind closing the door, please? Thank you very much. Okay, so here we see two representations of hexokinase. It has a molecular weight of about 100 kilodaltons, has two lobes, which actually swing together when glucose is being bound. So this is a relatively dramatic conformational change that this protein undergoes, which is shown better here if we actually compare the form where glucose is not bound and then the one where the substrate is bound. Okay. Occasionally compare this with this guy here, just to help you remember this. Now, glucose, the induced fit here that we see is a classic example of induced fit that was first described by Dan Koshland in the 1950s. I'm not sure if you heard about this kind of stuff. You probably heard about the lock and key model by Amy Fisher, which describes how a ligand and a biological macromolecule can interact with each other. In the lock and key model, based on Fisher, both actually already before the binding have a certain three-dimensional structure, which enables this steric fit also, so that we can have nice interaction between these two molecules. And in the 1950s, based on X-ray crystallographic studies, Dan Koshland then proposed that we don't typically have this rigid structure before a substrate is being bound by an enzyme, for example, or an antigen is bound by an antibody or any other ligand by another binding partner. But we have a conformational change that occurs when the ligand is being bound. So this conformational change, this fit that results from this conformational change, is induced by the ligand, and therefore this model is called the induced fit model. And nowadays it's known that pretty much in all interactions between such biological macromolecules and their corresponding binding partners, we have some kind of conformation. It can be very dramatic, as we've seen here for hexokinase, or it could just be the repositioning of some functional groups in the molecule. But typically, we have some sort of conformational changes occur. Sometimes it's more prominent, sometimes it's less prominent. So, the first step yielded phosphorylated glucose, which in the second step is converted to phosphorylated fructose. So glucose 6-phosphate is converted to fructose 6-phosphate, which is a simple isomerization reaction, okay, which is also implied by the name that we can give this enzyme. And there are a couple of names that you can find in the literature for this enzyme. The one that I'm going to use here is phosphohexose isomerase. That's what you also find in the textbook. Other names are glucose 6-phosphate isomerase or phosphoglucose isomerase. Never mind, they all catalyze the same reaction, namely to convert glucose 6-phosphate to fructose 6-phosphate and vice versa, as implied by the change in free energy, which is quite close to 0. This is a readily reversible reaction, and fructose 6-phosphate can also be converted to glucose 6-phosphate. So this step can also be used by gluconeogenesis when we actually produce glucose from starting molecules such as pyruvate. Okay. So all the steps that we discuss here in the context of glycolysis that are readily reversible are being utilized by the opposing metabolic pathway, gluconeogenesis. Okay. Those that are irreversible in glycolysis have to be bypassed in gluconeogenesis and replaced by other appropriate reactions. Okay. So the first reaction here that we discussed, catalyzed by hexokinase, is not reversible. So what happens actually, I should mention something here, in the reversal in gluconeogenesis is that an appropriate phosphatase cleaves off this phosphate to produce glucose. Now this reminds me that I should talk about naming here also. I always assume that Dr. Baker talked about these things. I hope that he mentioned kinases in 472, did he? And you know that kinases are actually phosphorylate stuff under ATP consumption. Okay, so for that reason, here we can appropriately call this enzyme that phosphorylates hexoses hexokinase. Okay, we'll see a bunch of other kinases from this class. Phosphatases, on the other hand, would cleave off such phosphates. Okay. So, as I said, this is a relatively straightforward isomerization reaction. And I promised those students who took my 470 class that we would take a closer look at some of these enzymes. And here is one, and I'm not quite sure whether we discussed it. I think this one I discussed actually, because I really like the simplicity here of the approach, the chemical approach that nature has taken to catalyze this interconversion. What you have to be aware of is that here we are dealing with an aldose, and here we are dealing with a ketose. So we have to convert a molecule that in the open chain form has an aldehyde at carbon number 1, or carbonyl at carbon number 1, to one that has a ketone or carbonyl at carbon number 2. Okay? That's all we have to do, but the chemistry that is utilized here, in my opinion, is actually quite ingenious, and this is shown on this slide. So as you may or may not be aware of, when we talk about enzymatic mechanisms, usually the active side of the enzyme, that's where all the action takes place, is outlined by an oval or line or something like that. And then we just see, often in an abbreviated version, amino acid residues coming from the enzyme that participate in the reaction one way or the other. Often enough, we don't even see the nature of the amino acid because either it's not known or it doesn't really matter. But sometimes the nature of the participating side chains is actually shown by using either the 3-letter symbol for the appropriate amino acid or the 1-letter symbol. Here's a 3-letter symbol. And we see that we have 2 amino acid residues are of great importance for the catalytic mechanism. I actually added these here. It's a glutamate here and a histidine here. Okay, so the way that the particular textbook that I got this from shows is that we only have B here and a protonated B here. B just stands for base. So if we protonate a base, we get an acid. Okay, if we deprotonate it, we get the base again. Now here, obviously, we are dealing with a protonated form of a histidine. Okay, so we have a positive charge here, and here we have a deprotonated glutamate. But okay, I think at the time that this was printed, maybe the nature of these amino acid residues wasn't known, or the textbook authors didn't really want to show this, or didn't really care. We have bound the substrate to the active side. It's still in the cyclic structure here, and then protonation of this oxygen here, the ring oxygen, facilitates opening up of this hemiacetal bond. I said this is readily reversible, the ring closure. At this point, we have not further derivatized this anomeric carbon, so we can do this. And we can convert the cyclic structure here to an open chain structure. Okay, so this is not shown in a straight chain form, doesn't really matter, but it's an open chain structure here. Now, not much has happened with regard to the conversion of the aldose to a ketose. Nothing really, okay? But now, genius strikes, because this deprotonated glutamate abstracts a proton here from carbon number 2. Okay, so this red proton is abstracted, moves over to our glutamate, forms a carboxyl group, okay, from the carboxylate. These electrons move over here, these electrons move over here, and we form a double bond between carbon 1 and 2, and an alcoholate here at carbon number 1. Okay, so we have a hydroxyl group, which is deprotonated, doesn't really matter. And both the position of this alcoholate here and this hydroxyl group here are on the same side with respect to this double bond. Okay, so what we formed here is an intermediate that has a cis positioning of these two functional groups with regard to this double bond, has a double bond, that we're going to call it an ene, and has two hydroxyl group, one of which is deprotonated. So we have a diol 8. 8 indicates we have a negative charge here, it's deprotonated. Okay, so this intermediate here, just by simply abstracting a proton here from carbon number 2, forms this, is the cis-ene diolate. That's all that we do at this point. Okay? Now what happens then is that this, at this point, protonated glutamate donates back the proton. Now, strictly speaking, it doesn't have to be the very same proton that was abstracted here from carbon number 2, so this can be exchanged with the medium. So if you label this one, okay, deuterium, for example, you wouldn't necessarily later find the deuterium in the now product. That's what's meant here. But a proton is donated back, but not to carbon 2, where we took it from, but to carbon number 1. Okay, just take it away here and add it there. Now, what happens is that we have now formed these electrons move over here, a ketone at carbon number 2, and we actually have here, after we close, actually we have the hydroxyl group here at carbon number 1. So we have converted the aldose that we started out with into ketones. Okay. Now we just close the ring again and we get the cyclized form of fructose 6-phosphate. So we take proton away, carbon number 2, and add it to carbon number 1. Have some electrons move around a little, and we have an isomerization of the aldose into the ketones. Now this, as we saw previously, is a readily reversible reaction, which also proceeds through this cis-ene diolate. And in my classes, my 470 students are not allowed to answer this question. But in my classes, I always ask what would happen, or what could also be formed in the reverse reaction starting with fructose 6-phosphate if we actually would not go only through a cis-ene diolate, but through a trans-ene diolate, where this hydroxyl group here would actually be on this side. So what would be formed here? We go from this structure, hydroxyl group goes up here, and then we form a phosphorylated aldohexose, but which one will be formed? Think about it. Okay, so what we have achieved so far is the production of fructose 6-phosphate, which in the third step is phosphorylated also at carbon number 1, okay, to form fructose 1,6-bisphosphate. So this is a bisphosphate, not a diphosphate. In a diphosphate, we would have the two phosphates linked to each other. In a bisphosphate, we have two phosphates actually found at different positions in the same molecule. Here, one is at carbon number 1, one at carbon number 6. Now this phosphorylation reaction also consumes ATP, so it's catalyzed by an appropriate kinase. And the kinase is called phosphofructokinase 1. Okay? So this particular reaction, which participates in glycolysis, which converts fructose 6-phosphate to fructose 1,6-bisphosphate, bisphosphate is catalyzed by phosphofructokinase 1, which implies that there is also a phosphofructokinase 2. Bear with me. Biochemistry nomenclature is sometimes confusing, but it's also often enough quite straightforward. You just have to remember it, okay? So when you want to talk about the particular enzyme that participates directly here in glycolysis, remember that it's phosphofructokinase 1. Now, in order to complicate matters, and there were many opportunities for biochemists to confuse biochemistry students, it seems, the phosphofructokinase 2 also plays an important role for glycolysis. But more precisely, the regulation of glycolysis, and more precisely, the regulation of this enzyme. Because phosphofructokinase 2, which we're going to discuss in a little bit more detail when we talk about regulation of glycolysis, produces fructose 2,6-bisphosphate. Okay, fructose 2,6-bisphosphate is the most important effector for phosphofructokinase 1. But again, let me take a step back. We'll talk about this in more detail once we talk about the regulation. But keep that in mind. This one is important. Now this reaction is also irreversible. It's not only hexagonic under standard transformed conditions, but also under physiological conditions. Okay, and this, as I implied already when I talked about fructose 2,6-bisphosphate, this enzyme is subject to regulation. Actually, a couple of effectors can regulate this. Activate or inhibit it. It's a kinase reaction. Okay, so we utilize the phosphoryl group, one of the phosphoryl groups from ATP, and produce ADP. Introduce the phosphoryl group here also at carbon number 1. So I'm not going to talk about this in more detail at this point. The fructose 1,6-bisphosphate still is a 6-carbon sugar. Be aware of that. And the 6-carbon sugar is now cleaved in the 4th step by an enzyme that is called aldolase, and there are different aldolases, class 1, class 2, depending on what you're looking at. So here we're talking about what happens in animals, okay? And it produces, this enzyme, actually 2 3-carbon compounds. So we use one 6-carbon compound, cleave it, and produce 2 3-carbon compounds. So stoichiometry is straightforward. And this step also gives glycolysis its name, the lysis of a sugar. Okay, it's not glucose anymore, it's fructose 1,6-bisphosphate that is cleaved. This reaction seems to be rather endogonic, but under physiological conditions actually closer to equilibrium, and therefore it's readily reversible under physiological conditions. So why do we call this enzyme aldolase? Because it catalyzes an aldol cleavage. Okay, and I'm not sure if you remember this from your organic chemistry classes, but an aldol cleavage is the opposite of an aldol condensation. And I like to explain these reactions starting with an aldol condensation. Okay. So, forgot to mention what is produced here, but we'll see these names again. This reaction produces dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. I'll talk about these again in a moment. But let's talk about aldol condensation and aldol cleavage for a little while, because this will help you to understand what actually happens here in this aldolase reaction. So an aldol condensation is a reaction between 2 carbonyl compounds. Typically, it's formulated in such a way that we have a ketone and an aldehyde. And under basic conditions, this ketone likes to donate a proton. Okay, so this is actually a carbon acid. Hope you remember this from organic chemistry. And due to the proximity of this oxygen here, this methyl group can lose a proton. Okay, under basic conditions. So what we form then is a carb anion, which we can also formulate here as an enolate. Doesn't really matter. I like to formulate this reaction with a carb anion. It's more obvious, I guess, because this is a very good nucleophile and likes to attack the partially positive carbon in the aldehyde. So if we do that, attack here, these electrons move out, and we form this intermediate, which after protonation forms this structure, which is an aldo. Okay? So aldol here. Now, what we see in the aldolase reaction is an aldol cleavage. So we just have to reverse this reaction. Also occurs under basic conditions. So what happens under basic conditions here with our aldol is that this proton here is abstracted. We form an alcoholate anion. These electrons can move over here. We can cleave this bond. Electrons move over here to form our first product, which is an aldehyde. Here it would be glyceraldehyde 3-phosphate. Okay? And a carb anion, which upon protonation actually produces our ketone. Our ketone here is dihydroxyacetone phosphate. Okay? So, again, I encourage you to remember the names of some basic 3-carbon molecules. Why is this molecule called dihydroxyacetone phosphate? Because it's a derivative of acetone. I talked about the structure of acetone before. It was a bisphosphorylated, not bisphosphorylated, it was a bishydroxylated acetone. It was a dihydroxyacetone, which now on one carbon has a phosphate, and therefore we call it dihydroxyacetone phosphate. Why is this molecule called glyceraldehyde 3-phosphate? Because we start out with a glycerol that was oxidized to an aldehyde, okay, glyceraldehyde, and now we have a phosphate here at carbon number 3, and therefore we call it glyceraldehyde 3-phosphate. Okay, so what we need in order to actually catalyze such a base-catalyzed enzymatic reaction is some sort of amino acid residue that can abstract protons. For the longest time, biochemists thought that this would actually be the phenolate anion of a tyrosine residue, and newer X-ray crystallographic studies then showed that it's actually an aspartate residue here, which abstracts the proton in the appropriate position, and then we have an aldol cleavage. Now, this is pretty much the same thing that we just discussed for the general organic chemistry here. It's just complicated seemingly a bit by the fact that the substrate is not just, let's say, floating around in the active site and it's held in place through non-covalent interactions, but that by the fact it's complicated by the fact that a transient covalent bond to another amino acid residue in the active site is formed. And we see this occasionally, that we actually have the reaction, covalent reaction with an appropriate amino acid residue that doesn't really participate in the catalytic mechanism, but its role is really just to hold the substrate in place. And the same is true here, because the substrate, actually the fructose 1,6-bisphosphate, which has a carbonyl at carbon number 2, it's a ketone, forms a Schiff base with an appropriate epsilon amine coming from a lysine residue. And this reaction between amines and carbonyls is spontaneous, it's readily reversible, and leads to the formation of a Schiff base. So this is a reaction in previous years I told my students, okay, that's one of my favorite reactions, and if I ask you what type of bond are we dealing with here, you don't know the answer, just say Schiff base, and often enough you will be right. So if we have reaction of a carbonyl, like a ketone aldehyde with an amine, we form this kind of bond here, okay, which is called a Schiff base, and it's readily reversible. So at the end, our second product, thank you for reminding me that it's time. The ketone will actually be just released after we hydrolyze this Schiff base again. But otherwise, this reaction mechanism is exactly the same as what I just discussed for the other organic chemicals. We have an abstraction of a proton here. These electrons move over here. We cleave the bond to this carbon, form the aldehyde which is released, which is the glyceraldehyde 3-phosphate in this case. And then we just have to hydrolyze, after some rearrangement, our dihydroxyacetone phosphate. So we convert fructose 1,6-bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and then we actually can convert these two into each other in the next step, which I'm going to discuss.